The overall goal of the following experiment is to increase the efficacy of the retrograde transport of neuroanatomical tracers to spinal cord motor neurons following intramuscular injection. This is achieved by initiating an acetylcholinesterase histochemical reaction in a mocked-up tissue of interest to identify the location of the motor end plates on the animal musculature. In the second step, intramuscular injections of the retrograde tracer are performed along the entire span of the motor end plate region on the muscles of interest. The animals are then kept for 14 days to allow the transport of the tracer into the spinal cord motor neurons. Ultimately, targeting the entire length of the motor end plate region yields more labeled motor neurons that extend across multiple segments of the rodent spinal cord when visualized using fluorescence microscopy.
The implications of this study extend towards the development of therapies for neuromuscular dysfunction, which includes spinal cord injury and amyotrophic lateral sclerosis, as this is a minimally invasive way and therefore a clinically relevant way to deliver therapeutic genes into the spinal cord motor neurons.
Generally, individuals new to this method will struggle because the motor end plate regions cannot be visualized in the musculature of living animals.
Visual demonstration of this technique is critical as the intramuscular injections require the mental transposition of the motor end plate onto the muscles.